Do you mind recording?
No. So I don't mind recording. I hope. Okay. So again, welcome to today's How to Interest Group meeting. Yeah, and without further ado, I'll let Greg present how to record to make an atlas with Figstream.
Yeah, thanks, Michael. Hi, everybody. So I've done several how-to presentations involving image registration before, and I thought I would kind of share what I think is one of the more advanced or complete applications of image registration, which is producing your own atlas. This is extremely common in the medical imaging field, where a group might be collecting MRIs of the human brain or the abdomen or the lungs from hundreds of patients. And rather than register each of those subjects' data independently to a public atlas, because just like in microscopy and medical imaging, there are public atlases that have manual annotations, data from different modalities, a lot of other useful resources connected to them. Instead of registering each patient's data individually to that public atlas, it's much more common to produce your own study-specific template. That is, to create an average image, or an atlas, if you want to call it that, of all of your subjects' data, and then register only that study-specific template to the public data set. So that way you kind of have public data set here, study-specific template here, and then all your subjects are registered to your study-specific template by default because you had to register them in order to make the study-specific template. A study-specific template is useful in a lot of other ways. If you want to create your own manual annotations that will be valid for all of your subjects at once, but you don't want to label them separately, then producing a study-specific template and annotating the template is a good idea, because then you can just propagate your annotations back to each of the individuals if you like. It's also useful if you're going to average results from all of your subjects together and you want to make sure you're averaging the correct anatomical locations to each other, or at least more correct than you would be if you just average the voxels naively without registering the data first. The first step for doing that would be to create a study-specific template, which involves registering all of the images in your data set to the same coordinate system. Okay, so I have slides. Those will take less than 5 minutes, I think, just to give you conceptually the idea of how we're going to make this template. And then I have a Jupyter notebook that lays out the code for making a template. And then we're going to make one right here in the how-to session using two-dimensional data for speed. But everything I'm going to show you works on three-dimensional data as well. And in BigStream, there's no difference to the function calls. This notebook will work just as well for 3D data as it does for 2D data. So let me start with the slideshow. Okay, so how to combine registered data to make an atlas. So let's pretend that each of these blue rectangles is an image you collected from the same organism, but they're different subjects. So you had 6 different fish, or 6 different mice, or 6 different flies, and you image the same structure in each one of those 6 experiments. But due to the images being collected on maybe different microscopes, or even the same microscope but on different days, maybe the images were stained slightly differently, and then most fundamentally, individuals just have variability in the size and shape of their brains, and in the specific locations of small structures like nuclei of clusters of cells can be in slightly different locations subject to subject. So that's what the sort of shading and the rotation and size of these rectangles is supposed to represent, the variability in your population of the anatomy. So to create an atlas from these 6 subjects, the first thing we're going to do is select one of those subjects to be the initial value for the atlas. All you're saying is, oh, you're making the simplest atlas possible. You're saying, all right, this subject is going to be representative of the whole data set. Then you register every other subject to that one individual. and then you use those registrations to resample the images from all of the subjects so that now they match that one individual subject as closely as the registration algorithm could accomplish. You could consider yourself done at this point and say all of my data is registered to the same coordinate system. But there's a few limitations with that. The first is that all of the individual subject images now look like the brain of that one individual. They've all been deformed or warped to match the brain of that one individual subject. And who's to say that that one individual subject is really a good representation of the average anatomy across the population for all of the subjects? The second limitation is that registration is imperfect. And so each one of these alignments has some residual mismatch where you have your initially chosen target and you have a subject registered to it. For that subject, some small part of the brain might be incorrectly aligned. For a different subject, a different part of the brain might be incorrectly aligned. And just using this first round of registrations to the one single target accumulates all of those errors. So instead of doing that, we're going to take all of these resampled images that roughly match the initial target, and we're going to average them together. So now I've moved the average back to kind of the center of where the original images all were. And it looks messy because, like I said, we just averaged all of those images together. And their registrations were imperfect, and they were, the shape of this initial template is biased to the shape of that one subject and all of the limitations I mentioned. So now that you have this sort of first round average, the next thing you do is register all of your subjects again, but this time to the average. So that's it. That's the whole procedure. But I guess the thing I should have said is that then you iterate that. So each sort of outer round of this process is register all subjects to the same target, average the images, then register all subjects to that average, and then average all of those images, then register all subjects to that average and average those images, and so on and so forth. Each round that you do this, the anatomy in the averaged image will gradually approach something which is equidistant in terms of its shape from all of the individual subjects. You are gradually moving away from the bias of registering all your data to one specific individual towards registering all your data to a common representation that is roughly equidistant in shape from all of the subjects in your data set. So let's summarize what I just showed you in pictures, but with sort of like text pseudocode or comments. Then we'll look at the actual code, which shows you how to implement this idea. So you've got a bunch of images from the same population. Choose a sample to be the initial value for the atlas. For n iterations, and this number you have to choose, although you can set some sort of automatic convergence criteria for this. You can say I want a maximum of n iterations, but if after n minus m iterations, the movement of the shape of the template is small enough, you can stop. So you can sort of decide my template, which I'm constructing, this average image, is no longer changing very much each iteration. You can stop. We don't have a nice early convergence in the example that I'm going to show you later, but it's easy to build. So register and then resample all of your individual images to the current atlas. Remember, the current atlas is initially on the first iteration, just one of your samples that you chose. Here's the averaging part. Histogram match all of the samples to the current atlas. I've been saying average the data a bunch of times. And what I mean by that really is after the data is registered, you just at every voxel take the mean value across all images at that, all registered images at that voxel. But you should do a little bit of signal processing first. If you just take the average right off the bat, that's going to be sensitive to differences in the level of expression of your fluorescent reporter, differences in the laser power at acquisition time, which is not always under your control. Anyone who's collected, fluorescent microscopy data knows that you can set the laser at the same laser power, experiment to experiment, and get totally different brightnesses in your images. because the laser has to heat up, the laser cools down, the power coming into the laser is also variable from the power grid, unless you use a fancy power conditioner or something. There's all kinds of uncontrolled variables here that we're not even aware of. So we want to computationally make sure that before we take the mean of the registered data, we are normalizing the histograms, the intensity values in the images, so that the mean makes sense to do. And histogram matching is a reasonable choice for this. So after you've histogram matched, average all of the registered samples. This average image is now the new current atlas. That's it. That's the whole algorithm. Choose a sample, register all samples to that sample, histogram match everything, average those together, do it all over again. A larger value for n will result in an atlas that is less biased to the initial choice of the target. The more sort of outer iterations that you do here, the more you are combining all of the subjects from your data set in order to determine the shape of the atlas that you ultimately are constructing. And like I said before, histogram matching accounts for variabilities in brightness due to differential expression of your fluorophore and the variance in the laser power, or anything else that changes the, that scales the histograms of the images that you collected. Okay, that's it for slides. Any questions about the general procedure that we're going to follow here before I jump into the code? Okay, sounds like we are doing good. All right. So I have a set. So there's a question. Yeah, go ahead.
So about sample speed, do you upsample the digital images? Because what I'm worried about is once you start doing rotations, you're going to lose Nyquist.
I'm having a little trouble hearing you through the Zoom microphone. I think you said something about upsampling and Nyquist sampling.
Okay, I found the microphone. How's that?
That's way better.
Okay. I was wondering if you do any upsampling before you proceed into this, just because I'm worried that if you start doing rotations or stretching, that you might lose the sample criterion, and so I'm just wondering what.
Interesting question. The first thing to point out is that once data sets are registered, when you resample the moving image to match the fixed image, you resample it onto the exact same grid as the fixed image. So at the time in which you average the data, all of the data sets have exactly the same number of voxels, the exact same voxel spacing, etc. But your question is a good one when you think about your initial data set. It's possible that you collected, most experiments like this, all of the samples were collected with the same imaging protocol. So usually they have the same voxel spacing, and that at the time in which that imaging protocol was designed, Nyquist frequency was talked about, and sort of what structures you're imaging was talked about. Are we imaging diffraction limited spots? Are we imaging big structures? What sampling rate do we really need? What SNR do we need? And so on. And usually those characteristics are the same for all samples, but not necessarily. Especially if you're a computer scientist doing this work and not a biologist doing this work. Often you're just downloading images from the internet and thinking, hey, what if I combine these? And that way I can combine data set A and B and study them at the same time. In that case, you might need to reconcile some differences in imaging protocol with respect to like sampling rate and things like that. You probably in that case would choose as your initial template whichever image had the highest sampling rate. So in that case, when you are resampling all of the other images, they're only ever being upsampled instead of downsampled when they're being resampled through the registration. There's trade-offs for that decision as well, because if you have one image that has tons of voxels and everything else with very few voxels, you'd be blowing your data set up very large with a lot of upsampling. And you have to think about space and computational complexity and time as well. But generally, you can get away with choosing as your initial template whichever image is the best quality, which usually means the highest sampling rate, the highest SNR, the least damage to the tissue, a sample that is, that already looks as best as possible like a representative example of the population. I think it was John who had thumbs up back there. I just want to acknowledge that at least at Janelia, John is the expert in this subject. He's produced all of the JFRC fly atlases and things like that. And there's more than one way to make an atlas. What I'm presenting is what I've done in my medical imaging work, but John might have insights on ways to do this that are specific for microscopy or other types of improvements that you could make, or tricks that would make this easier, faster, things like that.
And one more question for
Mhm.
histogram visualization. How do you deal with quantization?
So there's two different algorithms, and I want to make sure that we're differentiating them. There's histogram normalization, which is something you can do on one image by itself. Typically, that means you are stretching the image histogram to cover the full bit type of whatever data type you have available. So instead of just going from, say, 0 to 1,000, now your image intensities are stretched from 0 to 65,000 or whatever 2 to the 16th power is if you're using 16-bit data and so on. That's usually called histogram equalization or histogram normalization, I think, sometimes. So what I'm using here is histogram matching, and this requires two images as input. So you have a reference histogram, and then you have your the image whose histogram you want to change. And the way the algorithm works is it changes in a nonlinear way the histogram of one image so that the CDF, the cumulative density function of the two images, is identical. So it maps, say intensity A from image A, intensity 1 from image A to some other intensity from image B, such that the cumulative histograms of those two images are as close together as possible. And I think if I'm interpreting your question correctly, the question could be about that process is one where you have to bin things. You have to decide how many bins to use for your histogram. I don't put that much thought into it, to tell you the truth. I just use the default settings for histogram matching algorithms in scikit-image. I think that's usually 256 histogram bins. I think that if you use far fewer bins, you can run into some problems, or far more bins, you can run into problems in either case. But for the types of imaging that I usually, the relevant thing to think about here is, what does the native histogram look like? It ranges maybe from 0 to 8,000, and it has a specific shape. If I sampled that native histogram into 512 bins, would that still look like, to a nice approximation, the real histogram of the image up to the bit type precision?
I think you mostly answered my question. I think maybe just in a normalization case, for example, you were stretching from 0 to 1,000 to 0 to 65,000, right? If you just do multiplication to do that shift, I would expect intensity values every 65 values, right?
Mm-hmm, mm-hmm, mm-hmm.
And
and you don't have that much data, right? Your data is not actually available at that resolution. I think that what happens in that case is a lot of interpolating. So all of a sudden you get some values that shouldn't have originally existed in your data, a value between, most of these histogram normalization and histogram matching algorithms force a floating point value bit type. So a lot of the data we collect is integer valued, but scikit-image histogram match and scikit-image histogram equalization both convert the data to 32-bit floats first. And I think they do that because they resolve this quantization problem that you've mentioned by just taking, say you've got a bin, all the voxels that are value 100, they'll split that and then stretch it over multiple floating point values in order to keep the image looking smooth. And I typically allow the algorithm to do that, and then I round. But that's an important point, that you could subtly change the characteristics of some of your data by histogram matching. But remember, the output in this particular case is a new image, something which is an average representation of the shape and brightness characteristics of your population. You're not actually changing anything from your original raw data. And it's actually a good thing, this sort of stretching of intensities across the full bit type and interpolating between values, in the atlas image, because that might actually be more of an average representation of what you would collect on the microscope over multiple samples.
You mentioned interpolation. You usually use just linear interpolation.
I think for a lot of the like scikit image stuff that I use, it's available as an option. Like you can say order equals. Default, I'm assuming, because most of the NumPy SciPy defaults are cubic B-spline. But a lot of the times when I do this stuff on big images, I set order equal to 1 because it's way faster.
Okay, thank you.
Yeah, you're welcome. Okay, so we got into some advanced conversation about the histogram stuff. Let me get us back to the Atlas production stuff. So remember, what we want to do is take a collection of samples, choose one of them, then register everybody to that one example, histogram match all of that, average it, and then repeat. So I have a collection of 7 images of the zebrafish brain. These are 2D. I just grabbed a slice from the middle of the brain for all of these samples. And here's what they look like. These are all different fish. And you can see even as they load that the intensity characteristics are quite different for a lot of the different fish. Some of them are more bright in the forebrain. Some of them are brighter in the center than the others and things like that. You can also see shape variability between these individuals. They were all collected on the same microscope with the same acquisition parameters. But they were collected over months. Some of these were collected in 2022. Some of them were collected in 2023. And the population of these fish in the vivarium, the aquatic center, it's changing genetically over time. The type of food they're getting gradually changes. How the exact age of the fish at the time it was imaged might be a little bit variable by a few hours. There's a lot of things that contribute to why this fish is a little longer and skinnier, and this fish is a little more short and fat. And we want to register that out. We want an image that is an average representation of all of that variability, equidistant from all of the variability present in the population. Okay, so here's our Jupyter notebook that's going to accomplish this for us. This line, okay, we'll leave it. It's fine. First cell is just going to load all of the images. That's it. And I'm manually inputting the voxel spacing for these images. So remember, these are just 2D data for demonstration purposes and speed. And on the confocal microscope upstairs in our imaging protocol, it gives us a voxel spacing of 500 nanometers approximately in XY. And then this list comprehension is just reading each of the 7 images that are in this folder into a list. And then I'm going to print the shapes of each of those seven images so that we can make sure that they're, yeah, okay. We got 7 images, 7 2-dimensional images. Okay, so now this next cell is just going to define one function. And it's kind of the inside of the loop that we're going to run to create the atlas. So here's the pseudocode we had before. This function that we're defining in this cell is going to take care of these 3 lines. How do we register the data? How do we histogram match the data? Oh, sorry, actually just these 2 lines. It's not going to do the averaging. So it's just this function is going to do these 2 lines. So I've shown BigStream in how-to videos or how-to presentations before. This is parameterization of a 2-step image registration that performs an affine alignment followed by a deformable alignment. Those 2 steps are bundled together into a list or into this kind of steps list that BigStream uses. So we're telling BigStream, I want you to do an affine alignment, and then I want you to follow that with a deformable alignment afterwards. And here's where we actually run the alignment. So we're saying take the fixed and moving image. We know that they have the same voxel spacing in this particular case. And I want you to run these alignment steps on them that we defined here in this list. And that's going to return 2 independent transforms. T1, which is going to be an affine associated with the first step, and T2, which is going to be a deformable alignment associated with the second step. That's all been shown in how-to meetings before. If it looks unfamiliar, watch the how-to presentation on how to register your data to an atlas, your single image to an atlas. Okay, so when we have those transforms, we want to apply those transforms to resample the moving image data. So fixed and moving image, voxel spacings, and the transforms we want to apply are T1 and T2. That's going to give us a new image called aligned, which is the moving image resampled onto the fixed image. And then here's the histogram matching part. I'm just using scikit-image, match histograms. I'm saying take the aligned data and match its histogram to the histogram from the very first image that we loaded. So this is our images list that we loaded here in this first cell. I'm using the first image in that list, image 0, which will be this image right here, as the reference histogram for all of the images. So when I run this cell, nothing will happen because all I'm doing is defining a function. It should run quickly. It's done. And that's it. So now we have that function defined. Now, here's where we're actually going to start doing the work. So this third cell is going to account for this part, averaging all the histograms in each, averaging all of the registered data in each iteration. And it takes care of this sort of outer loop as well, sort of making sure that this happens iteratively multiple times. So we've got all we've got at this point is our list of images and what we want to do with each image pair. And so the first thing we do is we choose our initial atlas, which is just one subject from the data set. So atlas is a copy of the very first image that we loaded. And then I'm going to save that atlas to disk, because what I want to do as this is running, what we're going to do here is we're going to look at the atlas as it changes per iteration. We want to see what it looks like after 1 round of alignments, 2 rounds of alignments, 3 rounds of alignments. And we want to see that it is gradually changing size and shape to look like something that's more representative of the whole population instead of just one zebrafish brain. I just set a reasonable upper bound for the total number of iterations we want to run this. I print some feedback to make sure we know where we're at in the process. And then here's the actual alignment part. So if we're on the first so what we do on most iterations other than the first one is we run the function from the previous cell, which aligns 2 images and matches their histograms on the current atlas and 1 subject from the data set. And we do this for all subjects in the data set. So this list comprehension here is the register all subjects to the atlas part. On the first iteration, the only thing that's different is we make sure not to do this alignment for the very first image in the data set, because that's already the atlas. So if we did, say, align, resample, match histograms, atlas, and images 0, you'd just be aligning an image to itself, and that's a waste of time. So we skip that one. That's all that this if triple I is 0 part is for. Okay, and then for each round, this is the average part. So this is where I take all of the aligned images. So this is all the images aligned to the atlas and histogram matched to the same target. And I take the voxel-wise mean of all of them. I round that and put it back into 16-bit data type. And then I write that out to disk so that we can look at them. Now, I should have run this before I started explaining it. It's pretty quick because it's on 2-dimensional data. But we'll have to wait a minute or two before we get through with the first iteration in order to see how things proceed. Let's see, we've already got a new image in the folder, Atlas 0, which is just a duplicate of 0.nerd, the first image in the data set. So let me set this over to the side and make it smaller so that I can start opening images. And we'll keep this at sort of the edge of the algorithm. So here's our population that we're creating an atlas from. Now is also a good time for questions if there are any more about the code or about the data set or anything, now that you've seen this in a bit more concrete of an implementation.
This is a question like probably partly about the data set, which is like how much, if any, like all of these images look quite nicely like vertically oriented. And did you do anything beforehand to make
I rigid aligned everything first, just so that they would be, and I only did that because I wanted 2D data and I wanted to take just the same slice from all of the subjects. If I were doing this in 3D, I would not have rigid aligned everything first. I mean, that's part of the atlas construction, right? But because I wanted 2D data and like initially my fish are like, kind of all over because they're small and they're embedded in agarose. and the embedding is kind of hard, and so they settle in different orientations. And so the 3D images were rigid aligned, and then I cut the same slice out of all of them first.
Nice, thanks.
Yeah, yeah. That's a sort of a demonstration purposes only thing to do. You wouldn't have to do that on your own data set if you were doing it in 3D. Okay, so let's open, make sure this is still running, first of all. Yep, still running. We're on iteration 3, and let's take a look at some of the things that we have written out to disk so far. Let's take a look at the first 3 values of the atlas.
I have a question.
Sure.
So can you also perform this with multiple fluorescence labeling?
Sure. It just depends on, what you have. So what I'm showing, all of the samples are stained in exactly the same way. They're all from the same exact genetic line. Let's say you have multiple sample, like the most common thing to do to integrate this with like an experiment that you're doing is to make sure that for each sample you have at least one channel in common. So maybe you're collecting, 3 different channels per subject. One of them stains all cells, and then the other 2 stain the things you're actually specifically interested in studying, like a subpopulation of neurons or only cells that are positive for a specific gene or, something like that. So then you would use the common channel across all subjects in order to produce the atlas. That's the most sort of standard, reliable thing to do. You could do other things if you got creative. It's possible that if you had channels that had sufficient overlap in the amount of in the areas where signal was expressed, you could use different channels. What would happen is that only the areas that are common, the intersection of expression, would end up being present in the atlas in the long run because of the averaging. So you have multiple subjects aligned to each other, and then you're averaging all that together. Only one subject has this structure labeled, so that label doesn't show up with enough signal in the average, and so on.
Thank you.
Yeah.
Go on. There's another question in the chat.
Oh, sure. Sorry, I can't really see. Oh, I can't see chat. Should I just open it, or do you want to read it?
I can read it.
Okay.
Someone asks if it doesn't matter if in 3D images you have different number of planes.
No, it does not matter. That's perfectly fine. The thing that matters more than the number of planes in the image or the number of voxels in XY for that matter is that to the greatest extent possible, the same anatomical structures are present image to image. It doesn't really make sense to make an atlas from 4 images of a heart and 2 images of a brain. That's an extreme example, but the same principle holds for like, subregions of the animal you're studying. If I had, say, half of these images were only of the left hemisphere and then half images were only of the hindbrain, it doesn't make sense to make an average from those things. So again, like in the same vein as what I said about where the contrast agent or fluorescent marker is expressed, you can get creative with your own specific data set, even if you don't specifically adhere to the all images have exactly the same content in them principle. You can still make some of this stuff work with your specific data set. You just have to be more knowledgeable about what's going on. You're registering this, you're trying to register multiple copies of the same structures and average them together to produce something that is representative of the population. And as long as you feel that you can do that with the things that are resolved in your image, the things that are stained in your image, then yeah, different numbers of planes is totally fine. So when all of the individuals get resampled in order to match the atlas, at that point, everything is on the same voxel grid. So everything will get upsampled so that you'll have the same number of planes at that point, but for all samples. Okay, let's look at the first 3 iterations here, the atlas from the first 3 iterations. We have some more that we can look at in a minute. Remember, the first iteration, we initialized the atlas with the first subject. So these 2 images are identical. It's a bit slow, but I'm going to prove that by just putting these 2 images on top of each other really quick. It's a tautology. It's obvious that this would be true. But we're going to do it as a sanity check anyway, because at least I find that I am so prone to making silly mistakes when I do computational work that these sort of like obvious sanity checks sometimes do save me. But okay, the fact that it's all yellow is good. That means that 0, which was in red, and Atlas 0, which was in green, are identical images. So now the next thing I want to do is actually put Atlas 1 on top of it. So it's also, let's just qualitatively, there's already some differences between Atlas 1 and Atlas 2. Or sorry, Atlas 1 and Atlas 0. Atlas 0 looks like an individual sample. You can see individual cells resolved clearly. Atlas 1 doesn't look like that. It's a little bit fuzzy. And the reason it's a little bit fuzzy is because even if you have the large anatomical regions of the sample correctly registered, like the boundaries of cell body and neuropil regions, or glia and neuron regions, in this case, only cell bodies of neurons, only nuclei of neurons are stained. So these dark regions, that's neuropil or glia, so there's no signal there. So even if those boundaries are all correctly aligned, individual cells are never going to be in the same spot from subject to subject. They're different brains. They've developed along different trajectories. So individual cells are often not going to be in exactly the same spot. So even if you've done your registration really, really well, when you take your average, you're going to get some blurring in that area of where cells are specifically located. This is a good thing, not a bad thing, because that means your average or your Atlas image is now more representative of the population than it is of a specific individual. You are correctly capturing the biologically true notion that cells are not in exactly the same location from subject to subject. So let's put Atlas 1 on top of Atlas 0 and see how much things moved around. So things moved around. That's pretty much what I wanted to show, is that Atlas 1, which is in green, so the boundaries of the forebrain shrunk a little bit. The boundary of the tectum neuropil and the optic tectum cell bodies was pulled in a little bit. Actually, here you can just barely see, I'm not sure if it comes through zoom, that the hindbrain here is moved into the glia area a little bit. All of these things have changed because these locations in green of the anatomy are more equidistant from all subjects in the population than the original location of those same anatomical structures in your single subject. So we have moved the shape of the brain to something that is more representative of the shape of all brains in your population than just the shape of one specific individual's brain. Okay, let's compare Atlas 1 to Atlas 2. Let's do Atlas 1 first. Oh, wait, that's not even right. Atlas 1, that's not right either. Atlas 1, Atlas 2, I'll get there. There we go. That's what I want. Okay, much smaller changes this time around. You don't usually need too large a number of iterations in order to get an atlas that's pretty representative of your population. 3, 4 is usually good enough. It depends on how variable your population is. If I was registering data sets from zebrafish at age 4 through 8, there's a lot more shape difference between brains covering that range of days. But since all of these zebrafish were 6 days old, approximately, at the time the images were collected, there's not that much shape variability. So it doesn't take that much registering in order to find a single image that's a nice fit to the middle of the data set. But there is still some wiggling around a little bit of the boundaries. And the same thing will be true for the rest of these. So let me just pull out the last one. I think that should be the last one, Atlas 7, and compare that to Atlas 1. And we'll see how well, sort of what the difference, what did we gain from the final 7 iterations of the algorithm over just the first iteration. So not that much, a little bit of motion at the large boundaries. I would say it was worth doing these extra iterations for structures like this, where you have a very thin structure, these just like single layer of cell bodies or double layer of cell bodies was actually shifted to a new spot, that could be anatomically relevant for future studies, or, you know, it may be important to get that right. But I mean, that's pretty much it. I don't have a great conclusion, I guess, but that's how this all works. Once you have this image, I would take Atlas 7 here, and this would be my study-specific template. All of my individuals data, samples 0 through 6 up here, they are all registered to Atlas 7 by default. That was part of building Atlas 7. So now, if I want to average other pieces of data that I have from these, 7 subjects, I can use those transforms to put everything into the common template coordinate system and do that averaging. Maybe I have other channels like, staining of a specific set of cells. Maybe I have ROIs determined by functional imaging or transcriptomics or something like that. Now they're all registered to the same place where we have one image that's representative of the population. Another thing to do after averaging the data from all your subjects might be to take this Atlas image and register it to a public Atlas. Then you only have to do one registration, your study-specific template, register that to the public Atlas, and now all of your subjects are registered to the public Atlas by chaining together the transforms from your study-specific template to the Atlas and all of the alignments that you have of your individual subjects to your study-specific template. Okay, that's it. Are there any other questions about the data that we produced, the algorithm, the implementation?
Right, can you put this notebook on the wiki for how it's done?
Yep, the notebook and the slides, I'll put both on the wiki for how to. And I'll leave this data in the same location that's indicated in the notebook. So if you want to just try it right off the bat with the default data as a test, that's fine. I'll leave this is a small amount of data. I can leave it there for a long time. Eventually, I'll probably delete it, but like maybe in a year.
Do you have a feeling for how choices in like the registration parameters would affect how the Atlas changes over time? Like, I guess I intuitively feel like if the registration was, like if you were allowing more deformation, say, then maybe you'd expect more changes on later steps, but I'm not really sure. I don't know. What do you think?
Yeah, no, you're absolutely right. I chose parameters for the deformation here, or for the registration overall here. So the way I didn't go into them in detail because I've talked about using BigStream for image registration in previous how-tos, and I've talked a little bit more about what all these parameters mean in those presentations. But the choices that I made here were all meant to be fast because we're doing like multiple rounds of many registrations here, and I wanted it to show up in almost real time. But if I increase the spatial resolution of the deformable alignment and let it run for more iterations, things would probably move a little bit more. And I think what you would end up with is an atlas where the edges are a little bit more crisp. And they're all, remember what I said before about how like cells aren't always in the same place across different subjects. That's actually not always true. There are some cells that are close enough to each other that even across subjects, you could always register them together. There's 2 really big cells in the hindbrain of zebrafish. They're not present in this particular Z slice, but they're called the Mothner cells, and they control the final decision about whether a fish is going to swim or not. And they're huge compared to other cells in the zebrafish hindbrain. They're really big somas. And so those cells are so big and so often in sufficiently consistent locations that you can register them together in an atlas and have them show up. But your choices for how you do the affine and the deformable alignments would have to be good enough to make sure that the registrations done in each round could put the Mothner cells correctly on top of each other in each one of those rounds. But still, even at this lower resolution, we still have a pretty single cell resolution looking texture in the data, which means we haven't completely lost the sort of single cell nature of the individual samples.
Greg, I have a slightly, I like to apply this to a slightly different problem where, so I'm using a ball, so I have these flies and they're sitting on a ball and they move the ball around. And I want to track movements in the ball. So there's a video camera pointed on the side of the ball. I take images of the ball, video reflect them. Could this sort of workflow work for that, where I would map the layout of the ball, like the ball has shapes on it basically, so each image is slightly different as it moves. And then make an atlas of it and then register each change or movement in the ball to the atlas.
For each individual experiment, you're collecting sequential images of the ball, right?
A video, yes.
Yeah, I think that a better choice for that would be motion correction or optical tracking, which is related to this in the sense that you have a bunch of different images and you want them all to be co-registered. But the way you would do that one is probably register the first image to the first frame to the second frame to the third to the fourth, and so on and so forth, so that you're accounting for the delta in the ball at each time frame change. And actually, BigStream has a built-in tool for doing that. There's a whole module in BigStream called motion correction, and it's meant to take in either a 2D plus time or a 3D plus time data set and measure the amount of motion frame to frame across the whole time axis.
Okay, thank you.
Yeah, you're welcome. And actually, the 2D plus time feature in BigStream motion correction is new. It was originally just 3D plus time, and then somebody was like, asked me for 2D plus time, so I put that in. It didn't take that long. It was nice.
Okay, if there are no more questions, and we don't have anything else to say, Greg.
There was one other thing in the chat. Oh, just somebody saying thanks. You're welcome.
Yeah. Then thanks for the talk, Greg. And see you in 2 weeks.
Thanks, Greg.
Yep. All right, everybody. Thanks for coming.